This is the story of a simple, innocent cell that was plucked from its obscure but happy home and dragged into the floodlights of NASA's astrobiology program. Tragedy followed. Now, we all love the idea of space aliens. Biologists especially are keen to see how life could have been done differently. Sadly, space aliens haven't come to visit us, and going looking for them turns out to be very expensive. But maybe we can find alien forms of life here on Earth. All the life we know of has one common origin, but what if there were other origins and other alien descendants living unknown among us? How would we recognize them? Well, we could look for replicators, tiny ones maybe, living in obscure environments, places we'd never looked. We could look for differences in basic chemistry. Maybe they'd use silicon instead of carbon, or arsenic instead of phosphorus. So my story starts with an ambitious young researcher who was funded by NASA, who agreed that a shadow biosphere would be the next best thing to space aliens. She looked for the shadow biosphere in the sediments of California's Mono Lake, which are full of arsenic. She took sediment, mixed it with simple nutrients, but with arsenic instead of phosphorus, and she waited, and eventually it grew. Cells grew, and they kept on growing. So she pulled together a team of other NASA-funded researchers, and together they probed the cytoplasm of these tiny cells to showcase their special qualities. It turned out that these cells could grow in very high concentrations of arsenic. In fact, they grew better when arsenic was there than when it wasn't. The cells were full of arsenic, and most excitingly, these cells appeared to have replaced at least some of the phosphorus in their DNA with arsenic. So the researchers wrote a paper, and it was accepted by the prestigious journal Science. NASA went into full star maker mode, first sending out a titillating press release, and then calling a big press conference. Suddenly, our hero, small little obscure cell, was all over the news. But within a few days, doubts began to surface as other researchers had time to read the paper carefully. Basically, they were not impressed. They found a lot of problems, but there were really 4 big ones. The first was, maybe our hero, or our cells, weren't actually using arsenic to grow. It turned out that the medium was actually contaminated with small amounts of phosphorus. Maybe enough phosphorus to explain the growth that was seen. Second, our hero turned out to be not quite so glamorous as had been originally hoped. Rather than being an exotic prince from the shadow biosphere, DNA sequencing showed that its close relatives were just ordinary working-class halobacteria. Third, maybe the DNA didn't contain arsenic after all. It hadn't been carefully purified, so it's possible that the arsenic just hadn't been washed away and it came along for the ride. The biggest problem, however, was one that chemists had known for years, and that is that although arsenic can form bonds shaped very much like those of phosphorus, these bonds are much less stable, and that if you were to make DNA out of arsenic, it would fall apart in a second, whereas normal DNA will last for millions of years. So I decided to repeat the author's experiments to see if their surprising results might really just be due to contamination. I did this work partly as part of my campaign for open science. I want anyone who's interested to be able to see how science is really done. And so I have a research blog where I describe my own research. And on this blog, I posted every step of the replication process, all the failures, all the setbacks. I found that the cells don't actually need arsenic to grow, and that the observed growth is explained by phosphorus contamination. I found that the DNA was very stable, and in collaboration with a graduate student at Princeton, we showed that there was no arsenic in the DNA. So we wrote a rebuttal paper and published it both on Science, where you need to pay to see it, and on the Open Access Archive. Now, finally, what went wrong here? It wasn't one single failure, but rather a cascade of fail, where everybody involved failed to do an adequate job. The one thing that didn't fail was the process of science. The scientific record has been set straight.